Hello, my name is Maria Ramirez Roman, and today I would like to talk to you about the effect of alcohol on sleep patterns in Drosophila melanogaster.
So, there are many experiences such as shock or stress that affect the human brain in order to produce changes in behavior. And in this poster specifically, I want to talk to you about how drugs, such as alcohol, affect sleep and sleep dysregulation. Now, it has been observed before in human studies that the effect alcohol has on sleep varies. And in this case, it varies according to drinking behavior, whether the person is in the acute, chronic, or in the withdrawal phase. However, in general, at high doses, alcohol decreases total sleep, while at low doses, it increases it. In addition, there are many genes involved in alcohol responses and in circadian rhythms, and some of these genes are very important, such as cycle, TIM, and PER.
So, in order to gain more information about this mechanism, we want to study how alcohol affects sleep and what are the neuronal and molecular mechanisms are involved in this process.
We hypothesize that alcohol alters sleep patterns by affecting neurons that influence sleep. So in order to test this hypothesis, we use Drosophila melanogaster because it offers a unique set of tools that allow us effectively to study alcohol-induced sleep responses. Specifically, most of the genes of the fly has been identified, and we can do rapid mutant analysis with it. And also, fruit flies show alcohol responses, such as tolerance, dependence, and withdrawal, that closely resemble those of humans. In our first set of experiments, we expose ethanol vapor to flies using the chamber in panel A. This is the ethanol chamber, and it consists of an air pump linked to the regulators which we use to regulate the flow of air. These regulators are the cells linked to the containers that contain ethanol or water. And what we do with this circuit is that this circuit turns this liquid ethanol and water into vapor, and we expose flies to this vapor. And the experiment basically consists of 5 minutes of 1.5 liter per minute of water. And then we switch that water into ethanol. And we expose flies to ethanol until all flies are knocked down. And then we add 10% of the time it took for flies to not be knocked down of water. And for the control group, what we do is that we expose them to water. And what we see in panel B is that after the experiment, we place the flies in these small tubes that measure locomotor activity as the fly passes through this infrared sensor. And this is a Drosophila activity monitor system. It's the main system that we use to analyze sleep. So, in general, what we do is that we expose flies to ethanol in the experimental group, water in the control group, and with the use of the Drosophila activity monitor system, measure their sleep pattern for a period of days.
So, as a matter of review, what we do is that we expose flies to ethanol vapor and then measure their overall sleep patterns using the Drosophila activity monitor system, and we measure it for a period of 5 days. Now, in general, what we observe is this graph that talks to us about the total sleep of, in this case, female wild-type flies. What we observe here is that in the y-axis there's a total sleep, and in the x-axis is the time. The dark part of the graph is the night, and the light part of the graph is the day. The flies that were exposed to air are symbolized by these black dots, while the flies exposed to ethanol by this blue dots. What we see is that flies tend to sleep during the night, wake up just before lights on, sleep during the day, and wake up just before lights off. However, when we expose flies to ethanol, is a decrease in total sleep during the night and an increase in total sleep during the day. Now, looking more specifically at what happens each day, we observe that during the day, flies tend to increase their total sleep, decrease the number of fragments or the number of fragments that they sleep, known as the number of sleep episodes. They decrease the number of sleep episodes, and per those episodes, they increase the time they spend sleeping there. So they increase the number of mini sleep episode duration. Now, during the night, we see something very peculiar. We see that flies decrease the total sleep, increase their number of sleep episodes, and decrease the mean sleep episode duration. So that at a 24-hour interval, and in these 24 hours, the day and night analysis are included. we observe no change at all in total sleep, number of sleep episodes, and mean sleep episode duration. What we think is happening here is that the effect of alcohol during the day is balancing out the effect of alcohol during the night so that the fly maintains an overall homeostasis after being exposed to ethanol. So, as a matter of review, during the day, total sleep increases, number of sleep episodes decreases, and sleep episode duration increases. During the night, total sleep decreases, number of sleep episodes increases, and sleep episode duration decreases. And in an overall 24-hour interval that includes both day and night, there is no change at all observed in total sleep or other sleep patterns. And we think this is happening because what happens during the day is balancing out what happens during the night. So, in our next steps, we want to examine how ethanol affects sleep in the controlled and stable environment of an incubator. And this is very important because it has been observed that the effect ethanol on the organism is very, very, very specific and is influenced by many environmental factors such as temperature and humidity. It also allows us to study how sleep is maintained before the experiment, what we call a baseline sleep, so that we can later on compare what happens in sleep after ethanol exposure with that baseline sleep.
So what we want to do in later experiments is that we want to place the flies directly inside the Drosophila activity monitor and place this monitor inside the incubator and obtain the baseline sleep for these flies for about 2 to 3 days before the experiment. But this is tricky because, as you recall in our last set of experiments, we link the ethanol chamber to 2 vacuum tubes that contain the flies, and we expose the flies to ethanol vapor until they are knocked down. Now, we place the flies inside the tubes inside the monitors. And this means that the ethanol chamber is linked to 2 monitors. And in these 2 monitors, there's 32 tubes. So the airflow is largely diminished into these 32 compartments. So we need to figure out the right dosage and the right time that it takes for the flies to remain sedated after ethanol exposure. And we have done some preliminary analysis of this. And what we have observed after studying various exposure times and various doses is that flies diminish their locomotor activity after being exposed to 1 hour and a half of 1.5 liters per minute of ethanol vapor. So later on, what we want to do is expose flies to ethanol while they're inside the incubator inside the monitors with the parameters just described above, 1.5 liters per minute for 1 hour and a half, and observe the overall effect that has on sleep. And later on, we want to compare the ethanol concentration of the flies that were exposed to ethanol inside the monitors with the flies that were exposed to ethanol in the vacuum tube so that we have a deep understanding of how alcohol is affecting sleep. And we're doing this because we want to use this experimental design to effectively study the neuronal pathways and molecular mechanisms that affect alcohol-induced sleep behaviors. Lastly, I want to thank all that have helped me conduct this investigation in any way possible. My PI, Dr. Alfredo Gessis, other mentors that have helped me, like Dr. Agosto, the graduate students from the lab, the undergraduate students from the lab, especially NMC Sayana Santiago, and Sai for helping me funding this investigation.